I've been asked to talk about the current work on brassicas that we're doing as part of a much larger 3-year AHDB project on bacterial plant pathogens across a range of crops. So as I mentioned, the project number is CP191, or if you want to follow it on Twitter, it's hashtag CP191. And within this project, we're working on 2 bacterial diseases of brassicas. Black rot caused by Xanthomonas campestris pathovar campestris, which we sometimes abbreviate to XCC, and spear rot or head rot of broccoli, which is caused by particular biosurfactant producing strains of Pseudomonas fluorescens. So what are the symptoms of these 2 diseases? Well, Xanthomonas infection is commonly seen in the field as V-shaped yellow lesions with blackened veins, and these very much start along at the leaf edges. At its most extreme, this disease can kill plants and result in complete crop loss, but thankfully this situation is quite rare in the UK, and more commonly it causes reductions in yields through trimming losses in winter cabbage, exposure of curds in collies through the loss of wrapper leaves, and direct losses of marketable product in leafy crops like kale. Spear rot, as its name indicates, causes a soft rot of mature broccoli heads, both in the field and sometimes soon after harvest. So what's the thinking behind this project? Well, of course, we know that ideally growers would like to have an effective spray to control bacterial diseases. But despite many trials over the last 30 or so years, we've failed to find any really effective alternatives to copper sprays. And the use of copper is now very much restricted. So the basic theme behind the project is that the best way to control most bacterial diseases is to keep them out or prevent them being introduced onto the farm in the first place. And to be successful, this means we need to understand the epidemiology in order to target effective control measures, and such as by setting health standards for seed and propagation material. So for Xanthomonas, we did a lot of work in the past on the epidemiology. And this showed that the main problem was very rapid spread during plant raising from very low levels of seed infection. And this led at the time to the proposal of much more stringent seed health standards. Over the last few years, though, there seems to have been a resurgence in the disease, and we're trying to figure out why. Firstly, we're doing, I guess you could call it a track and trace exercise with a particular grower to try and confirm where the disease is coming from. This is involving testing plants in the field, testing the seed, and testing the transplants as they arrive on the farm. Second, we're planting out specially raised high health transplants, and these we expect to demonstrate the role of transplants or not in bringing disease into the field. And thirdly, we're working with plant raisers to look at the feasibility of a radical alternative plant raising system, which eliminates the conventional overhead irrigation, and in turn reduces the rate of spread of the pathogen during plant raising. So what about the work on broccoli? Well, for spear rot, we have some evidence from work done more than 20 years ago that there are differences in susceptibility amongst varieties, but there's been no independent testing of recent varieties. So we're running a trial hosted by East of Scotland growers to see if we can pick up differences between varieties. The transplants for this trial are inoculated with a known strain of the pathogen, so that we can be sure that any difference we see in the field are due to inherent differences in resistance rather than whether or not they're carrying the pathogen. We're less certain about the epidemiology of spear rot than we are for black rot. And again, in previous work, we had indications that the pathogen could be present on the seed, transmit to transplants and then survive on the plants up until heading and cause disease. And if this is the case, then there may be potential to control the disease by reducing pathogen levels in the seed and reducing spread in transplants using the same approaches as for Xanthomonas. So the first thing we're doing is we're testing seed of different varieties, that's seed from different companies, and in some cases multiple seed lots of the same variety produced in different countries. Secondly, we're looking at the rate of spread during transplant production, and we're also planning to use the same sort of system of high health transplants to demonstrate the importance of different inoculum sources in the field. Finally, I should also end with saying that if any growers want to get directly involved in the project in any way, that my contact details are on the website planthealth.co.uk, or you can contact Dawn Teverson at the AHDB and she'll be happy to put you in contact with me, I'm sure. Thanks.